what the overall process looks like for a patient is basically go to a doctor, they take a cell sample, basically just a biopsy, and then they ship it to OVEL, and we will reprogram those into stem cells. We'll do our differentiation protocol and ship eggs back to an IVF clinic that will then use them for combining with sperm and making babies from that. This is an alternative to egg retrieval, which is expensive, requires a lot of hormone injections, it is very time consuming, and actually it just doesn't work for many people who use it. So this will provide a more streamlined alternative approach that can also produce arbitrary quantities of eggs.
Welcome to Galaxy Balance, a podcast exploring the frontier where biology, technology, and exploration intersect. Each episode is a deep dive with scientists and builders shaping the long-term future of life, from aging and genome engineering to space biology and intelligence itself. The goal is to understand not just what is possible, but what actually matters as these technologies accelerate. Today, I'm joined by Merrick Smela. Merrick is a scientist and engineer working at the intersection of biology, computation, and human reproduction. Merrick recently completed his PhD in George Church's lab at Harvard, where he developed synthetic biology approaches to generate ovarian cell types from stem cells, foundational work for building human gametes in vitro. He is co-founder and chief scientific officer of OVEL, a biotechnology company focused on addressing infertility through in vitro gametogenesis, or IVG. OVEL's goal is to develop new ways of generating functional human gametes from stem cells with the aim of expanding reproductive options for people facing infertility. Roughly 1 in 7 couples struggle to conceive, and existing treatments such as IVF can be invasive, expensive, and often unsuccessful. IVG represents a potential next step beyond IVF, one that could be more effective, less physically demanding, and more accessible over time. Merrick brings a systems-level perspective to this challenge, thinking deeply about developmental fidelity, safety, and ethical responsibilities that come with working at the frontier of human reproduction. His work sits at the intersection of fundamental biology and real-world impact, with implications not just for fertility care, but how we understand development, aging, and biological information more broadly. Merrick, welcome to Galaxy Balance.
Thanks for having me, Corey.
Excellent. Looking forward to the discussion. I thought we'd start with a little bit more about your story. Could you tell us who you are, what got you interested in this topic and science more broadly, and some of the challenges to get where you are now?
Yeah, so I would say it all started back when I was in college. This was 2016, 2017. At the University of Minnesota, I was studying organic chemistry, basically making different types of molecules using new chemical reactions. But I was reading a lot of, I guess, like science, also some science fiction, and came across some very interesting research that was coming out of Japan about being able to make mouse eggs from stem cells. So this was a team led by Mitanori Saito and Katsuhiko Hayashi, who had succeeded in the first demonstration of mouse eggs grown in vitro. And I was also hearing a little bit at the same time about some of the early embryo selection companies like Genomic Prediction, and just the potential I saw was there for this technology to expand reproductive options, both, for example, for like older women, as well as synergizing with some of these other things like embryo selection that were in the pipeline at the time. One of my friends, Anthony Tabbitt, had recently been awarded a Churchill Scholarship to study chemical engineering at Cambridge University in England. And I noticed also there was a lab at Cambridge, Azim Sarani's lab, who had at the time published one of the first methods for human primordial germ cell specification from stem cells. So primordial germ cells are the very earliest stages of egg and sperm development, happen like right at the beginning of the embryo. And they were one of the first labs to be able to do this from stem cells in vitro. So I thought, why not also do a Churchill Scholarship there? So I put together this application. It was actually quite a lot. Like this is a pretty selective application process, but I was fortunate enough to be awarded the scholarship. So after I graduated from undergrad, I switched fields from organic chemistry to germline biology, stem cells, things like that. I've maintained the trajectory ever since. But during my one-year master's program in the Serrani lab, I studied transcription factors and their roles in specifying primordial germ cells. I basically was able to deplete levels of a certain transcription factor called SOX15 and examine the effects on the primordial germ cell specification. It turned out not to really be very impactful for that cell type, or at least during that time period, which was only a few days because that's as far as we could go at that point. But it was still an interesting experience in my first sort of touch with stem cells, and it sort of got me hooked in the field and made a lot of good connections there as well, because Azim Serrani has basically trained, I would say, like half or more of all of the scientists working in this area can trace their lineage back to him. Like his lab was the first one to show that imprinting was the result of DNA methylation, for example. They sort of co-discovered genomic imprinting back in 1984. So after completing my master's in the Serrani lab, I came back to the US. I joined George Church's lab, where I briefly met Corey. And there I continued working on the role of transcription factors in ovarian development. My first project was about ovarian supporting cells. I was working with another student, Christian Krem, who was also interested in this area. And in this project, what I did was identify transcription factors that could drive stem cells to become ovarian supporting cells called granulosa cells. These cells are important for hormone signaling, for example, producing estrogen. They can also support egg development. So I was able to show that I could generate these cells from stem cells in actually only 5 days using a combination of transcription factors. And one of the things I learned was that it's important to do this sort of screen in a way that makes sense, which is to have a target that you're optimizing for that is the target that you actually want. So what I mean by this is that if you have your readout of your screen being like correlated with the cell type that you want, that is often good, but sometimes can lead you in the wrong direction. You basically have to do your screen in a system that matches your desired output as closely as possible, so your screening hits are going to actually be real. A lot of previous efforts in the area had used systems where they got some aspects of ovarian cell function, but not quite the full thing. And I think part of the Church Lab technology that is so powerful is that we can sort of engineer these reporter cell lines that will, for example, become fluorescent if they express a marker for ovarian cell identity, and then do sort of an unbiased screen of huge numbers of combinations, like up to millions of combinations of different transcription factors, and pick out the ones that get us to where we need to go. So the first half of my PhD was about ovarian supporting cells, and then second half of my PhD was about meiosis. This was after sort of publishing the supporting cell project. And now meiosis is the process where the chromosomes will pair up and recombine with each other. So you inherit one copy of each chromosome from your mother and one copy from your father, right? So each cell in your body has two sets. This is known as diploid cells. So if the diploid cells are going to need to transmit one chromosome to the next generation, well, how do they do that? The answer is meiosis. So the copy inherited from the mother pairs up with the copy inherited from the father, and then recombine to generate like basically a new chromosome copy that then gets passed down to the child. And at the time, nobody had been able to figure this out or get it working from human cells in vitro. So I took a similar approach philosophically to my previous project, which is basically figure out candidate factors for driving meiosis, and then also engineer a system that will tell me when meiosis is happening, and screen huge numbers of combinations. I think we got up to like 600,000 plus cells in this one single cell screen experiment, and figure out which of these factors could drive meiosis. Now, here there were some interesting things as well, because it turns out a lot of what drives meiosis is actually not transcription factors, it's RNA binding proteins. So including those in my screen as well was crucial for the success of the project. So thankfully I was able to sort of predict which factors were likely to be important. I identified 3 of them that each individually could activate meiosis, and this paper was published in August of 2025, basically, although it was preprint was out a little bit earlier than that. So with his success at initiating meiosis, at the time I was trying to decide what I wanted to do after my PhD, and got talking with a few people, was trying to figure out if I wanted to do academia, if I wanted to work for another company, or if I wanted to start something of my own. A lot of people at the Church Lab had experience with founding companies, and actually I was contacted by my co-founder Travis, who at the time was working on Wall Street, and basically he had been having some like personal issues with fertility with him and his wife. He got interested in this area of research. He actually reached out to me first in 2023 about the ovarian supporting cell paper. So we had been in touch off and on for a while, and decided, we decided it was the right time to start an IVG company. So IVG being in vitro gametogenesis, growing eggs outside of the body. We thought the technology was not quite ready yet, but close enough that with a moderate amount of work, we could get it over the finish line into a point where it can actually benefit patients. So since then, we started a research lab. This is based at the Harvard Life Lab, which is a startup incubator in the Harvard Medical School campus. And we have been continuing our research. We've made substantial progress past what I've been able to do in my PhD in things like ovarian supporting cells with a more early stage signaling environment to support meiosis. We've done, more progress in meiosis, and then also progress in ovarian follicle culture, where we can take early stage ovarian follicles and basically grow the eggs to a large enough size that they can be then used for IVF. So that's what, I guess, it's turned into a bit of a longer story, but that's sort of my current path, and I'm excited to continue this research and benefit the millions of women who want children but currently aren't able to have them.
A very exciting story and path. I think one interesting part of the regulatory process for stem cells is that a lot of targets, they don't need to replicate the cell perfectly. For example, dopaminergic neurons, they really have this main function of producing dopamine. They aren't as interested as necessarily making all of the functions. Another one might be CAR T cells. You don't need necessarily the full function of it if it can present and kill the cancer cells of interest, but with meiosis and ovarian cells, more than others, it seems you have to get the full functionality to be therapeutically beneficial. As you mentioned in your screens, you get what you screen for, and you
Yeah.
have to screen for as close as you can towards that target cell type. Could you dig a little bit further of how you mimicked that or got closer on that first high throughput screen?
Yeah, so in terms of like required function for our cells, right? We need to be as close as possible to natural eggs. And I would say that is defined as eggs do have a range of natural variation. So as long as we're within the range of variation of normal eggs, that's fine. And we want eventually to be higher quality than normal eggs because actually only about 30% of eggs that are fertilized are able to develop to a healthy baby all the way. Um, and that number goes down even more with advanced maternal age. So the way we make sure that we're getting the right thing, it's sort of like there's a trade-off, right? You can do like a readout that is, I would say, medium fidelity, but you can do like a lot of, like, it's easy to screen like a few million factors at once, right? Or you can do a readout that's very high fidelity, but less throughput. So in terms of that screen in the Science Advances paper, right? So initially, this was just based on fluorescent reporter expression. So engineering cells to become fluorescent if they express like one or two genes, basically. So we can do multiple colors. Let's say if we have cells that are along the germ lineage, they turn red, and if they're actually doing meiosis, they're both red and green, right? And we can sort those out. And that lets us do like tens or hundreds of millions of combinations to identify factors that can get us like at least close to where we need to go. Beyond that, we can actually use single cell sequencing technology to get a fuller view of the global transcriptional state of the cells to see if it matches up with the cells in vivo. So that was the single cell screen I was talking about in the meiosis paper. We used the fluorescent reporters, but then we did additional sequencing of the sorted reporter positive cells to figure out, like, okay, of the cells that are vaguely in the right area, which ones are actually like matching up exactly with the gene expression of the cells in vivo, right? And that let us narrow down the list of factors quite a lot. And in fact, the factors that came out of that screen were like actually the correct answer. We had to do only minimal validation afterwards. Now, the thing is that this is true for meiosis particularly, right? There is a lot more to meiosis than just gene expression, right? Meiosis is a physical process where the chromosomes have to pair up and recombine with each other. And you can't tell, like, physically where a chromosome is in a cell based on just the RNA content of the cell. So more recently, what we've been working on is microscopy-based screens. So being able to do high throughput automated imaging and see, okay, like, what are the chromosomes actually doing? Are they pairing up correctly? So that is lower throughput for sure than this sort of single cell sequencing technology, but it directly gives us what we need in terms of the localization and conformation of the chromosomes. Obviously, it doesn't give as much information on the RNA expression, but we're at the point where we have that part down already. So we're basically looking now at, like, how are the chromosomes moving around? Are they pairing correctly? Things like that. And I think microscopy in this case has been quite helpful. And we have a system now where we can do automated screening of like 96 well plates, slides, etc. We can also look at protein markers in a few different channels as well, but it's sort of like a process of finding the right screening tool for the scientific question you're trying to address.
Interesting. And one thing I've seen from Perturb-seq and other high throughput screens, especially bulk, where you put them in together, is you often don't see full diversity. Certain members tend to predominate. You see much higher copies of that. What type of diversity did you see when assessing millions of cells?
You mean in the Science Advances paper, or?
In any of your high throughput screening work?
Yeah, well, I think the diversity depends on the quality of your starting materials. And if you have a library with low diversity going in, that's what you're going to see. But I think with, I mean, we're not like, I think GC Therapeutics is a lot more expertise than OVEL in designing these sorts of libraries and things. But we've been able to make our own libraries as well. And I think we haven't had that much issue with like certain things predominating over others. I think there was this one time where we were coming up with a hit that turned out just to activate one of our fluorescent reporters without
Mm.
driving the broader like changes that we wanted. But again, like that was a case of you get what you screen for, right? And we were able to work around that just fine. But I mean, does that answer your question?
Yeah, I think so.
Okay.
And let's move more into OVEL. You mentioned a great story of your founder going through personal fertility issues. It's a great motivation. What made you think that now is the time to address it? You mentioned it's still a little early, but you think you're within striking distance. How was that assessment, and how did you make that pitch to investors to get this started now?
Yeah. In terms of like doing OVEL versus doing other stuff, right? The main other thing I was considering was like applying for professor positions, working in academia to push this forward as like in academia. And I was also maybe considering working for another company who was in this area as well. But generally, I wanted both like scientific independence and then also resources and the ability to build a team of really exceptional people. And in academia, right, I think it's a bit limiting in terms of the like types of organizations you can have. You get grad students, maybe some postdocs. They might only stay for like a few years. If you're hiring grad students, they don't come in with the expertise that you're looking for. They have to learn as they go. Um, and I think like the technology is close enough to the point where we can think about doing clinical trials that, I mean, you basically can't do a I mean, it's very challenging to do a clinical trial out of an academic lab, I will say, because the system is not set up for that. So that's sort of why I decided to do OVEL, because I thought it was the right time. The technology was maturing, right? It's been this year, it will be 10 years since the first mouse in vitro oogenesis. And the human technology has come actually quite a long way since then. And I think the rate of progress has only accelerated in the last few years. Um, so I think like if I had done an academic lab, I would sort of be watching from the sidelines as other efforts were the ones to bring it to the clinic. And I think that this is the best way to move quickly and actually help the patients who need it.
Hmm. And to dig a little bit more, what's your vision for the therapeutic product? Could you walk us through at a high level for the audience, just from taking material from the patient or the donor all the way to having an egg ready for IVF? We've talked about your paper a little bit, the discovery side, but what's that final clinical product look like in your mind?
Yeah, so the process of in vitro gametogenesis or IVG starts with a cell from an adult. So we'll want to choose a cell type with a low mutational load because any mutations that are in the starting cell type will be present in the child. I mean, unless we can do embryo or unless we can do cell line editing to correct them. But at this point, that's not going to be legal in the U.S. So choosing a cell type, for example, like a bone marrow stem cell, which has like a relatively insulated environment, low mutation load, we actually have estimated that if we take about 5 different bone marrow stem cells that and choose the one with the lowest mutation, it will actually have lower mutations in natural reproduction in terms of the mutational load. So starting out, you take a cell sample from an adult, reprogram that into stem cells. So this is the induced pluripotent stem cell technology. It's been widely used in the field for several years. It won the Nobel Prize in 2012. And from those iPSCs, basically the next step is to push them to make eggs, right? So we can generate egg precursor cells that are doing meiosis. We can also generate ovarian supporting cells that will support the growth of these eggs. And then it's a matter of culturing them in order to get fully mature eggs out the other side. So what the overall process looks like for a patient is basically go to a doctor, they take a cell sample, basically just a biopsy, and then they ship it to OVEL, and we will reprogram those into stem cells. We'll do our differentiation protocol and ship eggs back to an IVF clinic that will then use them for combining with sperm and making babies from that. So this is an alternative to egg retrieval, which is expensive, requires a lot of hormone injections, it is very time consuming, and actually it just doesn't work for many people who use it. So this will provide a more streamlined alternative approach that can also produce arbitrary quantities of eggs, because you can grow stem cells indefinitely. You can make, like, as I said, as many eggs as you want, limited only by like cell culture media costs, which are actually a lot cheaper than costs of like IVF medications. So over time, we think this will be adopted quite widely as an alternative to egg retrieval for IVF.
Excellent. And I know one of your goals on the website is to democratize and make this available to all and reduce the time and cost. One big challenge seems to be the stem cell reprogramming part. At least the field generally to get GMP quality iPSCs is a big time investment and a big money investment. How do you see addressing that for your therapy?
Yeah. So the interesting thing, so for listeners, GMP refers to like good manufacturing practices. This is a set of FDA regulations that basically govern how labs are set up, how like what paperwork needs to be filled out in order for you to sell your product, right? The actual cost of stem cell reprogramming is actually not very high. I would say this is comparable, like at scale, to something like an mRNA COVID vaccine, right? It's sort of a similar technology where you put in a transient like RNA. You can also do non-integrating DNA expression into your cells, grow it. Eventually, this will reprogram them into stem cells. If we look at the actual cost of like just the materials and sort of basic equipment, we get a figure in like, I would say less than $1,000 to do a stem cell reprogramming. And I think that could even be lower, right? Now, if you look at the cost of GMP compliance, that's like on the order of 100,000, right? So it basically boils down to what regulation is in terms of like record keeping, quality control, things like that. And I think part of the reason it's high is that you pay the regulatory compliance costs as sort of a fixed cost. And the fixed cost is like quite expensive to just get the certification. And then after that point, like each additional cell line also requires like some amount of like regulatory compliance work in terms of like documentation, staffing, things like that. Um, the point I'm trying to make is I think that intrinsically we can make stem cells and do quality control for less than the cost of a traditional egg retrieval cycle, which might be like 20,000, right? I think like in terms of GMP, right? Um, we want to have a product that's trusted, and we will do all the quality control that we need in order to get that up to and including complying with GMP certifications. I think a lot of that can be spread across the cost of doing multiple cell lines so that once we're operating at scale, the cost will come down to closer to the level that it will be to just in terms of like materials costs. If you look at other companies like, for example, Celino, they are doing sort of the same thing. They have like an automated GMP compliant iPSC reprogramming, right? It's possible that we'll end up partnering with one of these other companies for the actual like iPSC reprogramming expertise, just in terms of like what makes the most business sense. But I think like when we're thinking about very large scale things and things that are in, let's say, 15 to 20 years, if we're doing like a multiple digit percentage of all the births in the US from this process, I think the cost of compliance can be spread across multiple patients, and that will bring it down, basically.
Yeah, totally agree. The actual reprogramming step itself, very low cost. But it's not just
Yeah.
paperwork and burden, but the QC required to do it and the facilities, as you say. That's often a one-term cost. And I do think there's a way to get there, and companies like Celino are definitely leading the way in that regard. But I'm glad you're open to outsourcing that, because I know it's a big challenge for a startup to do the the later step, but also just the reprogramming. It would take an
Yeah.
entire company of your size to address that.
Yeah. And I think also in terms of quality control, our goal here is actually to make eggs and embryos that are sort of higher quality than natural, right? Because as I mentioned, most of the eggs that get fertilized naturally actually don't develop properly. Um, and as a result, I think at the end of the day, most of the costs of our procedure will actually be in terms of quality control. So, for example, like embryo biopsies, sequencing, epigenetic analysis. Um, I think the actual like physical materials going in are not going to be very expensive at all. It's basically nutrients and like mRNA. So what does get expensive is like all the sequencing that we'll need to do for the embryo quality control. But again, I think we can get this to likely less than $1,000 per embryo at the end. And compare this with traditional IVF, which is like multiple tens of thousands per cycle, and cycles might not even be successful.
Yeah, and once you get there, how do you know that it's functionally competent? Other therapies, you often go to animal studies, but that doesn't seem practical with the egg. What's your plan once you get to the point that in vitro it looks competent? How do you prove that next level of functionality?
Yeah, so in order to develop normally, an egg needs three things. Number one, it needs the right genetics, so the right number of chromosomes, like no DNA damage, no harmful mutations. Second thing is epigenetics. So these are modifications to the DNA or histones that will control gene expression. So an egg needs to have the proper epigenetics in order for the resulting embryo to have the right gene expression and develop normally. And then the third thing is the right cytoplasm. So things like number of mitochondria, amount and quantity of RNAs and proteins. And actually, those three things are both necessary and also sufficient for healthy development, right? So there are a few things we can do here to make sure our eggs are good. Before going to humans, non-human primate trials will show that our technology can give healthy non-human primate babies. And then also on the human side of things, we can basically do like embryo biopsies, compare the cells with natural embryo biopsies, basically see if our cells are within the natural range of variation of um, embryos, even potentially from the same parents that are either like IVF derived or IVG derived. And we can see like, okay, are these cells developing normally, at least before implantation? And I think between non-human primate safety data and embryo quality control data, we can be quite confident before we go to human trials of whether our technology is safe or if we need to make changes to get it working better.
And are you close to testing conception with this? I've seen going through meiosis, getting to the egg stage. What point do you envision actually testing in vitro conceptions and getting to the blastocyst?
Yeah, so I think we're not at that point yet. We are hoping that, like, pending our current funding raise, we'll be able to get that, like, first demonstration egg in roughly 2 years, I would say, based on our current rate of progress. Um, and I will mention that we would have to do this outside of the state of Massachusetts, which does not allow creation of embryos for research purposes, at least for human embryos. But at that point, I think we would set up a lab in another state that would allow this for research purposes. So things like New York or potentially Oregon, California. Um,
Yeah, interesting. And you mentioned NHPs. I think that's a great step. Are you going to do rodent, mouse, or rat before then?
We're actually not planning to do any rodent work, and there's a few reasons for this. Number one is I think the field has realized a lot that rodent biology is actually pretty different from non-human primate or human biology in several aspects of stem cells and germline development. And things like ovarian follicle culture, even like methods have been developed for mouse for decades, but the same methods just don't work in humans. And I think if we were optimizing our method for mice, that would be a strategic mistake, I would say, because we're going to end up in a situation where we have a method that works, but when we try to translate it, it's not going to do it. We are doing other species besides like humans, right? So non-human primates are one, um, and I think like we can learn a lot from animal models, but I think mice and rats are probably not a good choice here. The one exception is that if we're required to do this by FDA as some condition of starting human trials, then we probably would. But we think that the non-human primate data will be enough for showing that our method is safe.
Yeah. So, what are the key challenges or key failure modes you see from where you are now to getting to that point of a ready egg for the next step?
Yeah, I think a lot of it is um, like just improving efficiency of technology that we already have in terms of things like follicle culture, being able to go from like a primordial ovarian follicle to a fully grown egg. This is something that's possible but hard and not very efficient. And if we start from a stem cell derived follicle, this will probably not work as well, at least starting out. Um, Also, things like epigenetics. We need to get so we've actually made a lot of progress on epigenetics over the last year. I think if you look at the epigenetics in the Science Advances paper, they were not very good, and I think that's why the meiosis in that paper was not progressing all the way. And by not very good, I mean, at the time, it was good for, at the time, I think it was better than most other epigenetic efforts, but it still wasn't good enough to match what happens in vivo. We've been able to get epigenetics like much closer to in vivo, where like at the point now where our cells are within the range of epigenetic variation that happens naturally, which was not the case before. But in terms of like maintaining epigenetics over the long follicle culture period, I think that's something we need to work on. Yeah, there's a few other like things that we've identified that I'm not ready to sort of discuss, but in terms of the overall roadmap, I think we have a pretty clear path forward in terms of getting our cells to make an egg. Like we've put together a lot of the pieces like as individual steps, and now we're at the point where we're sort of stringing different things together in order to make an end-to-end process.
Interesting. And I want to go into a little bit more about DNA damage and integrity. You mentioned starting from the bone marrow. If you pick several, you can get within that normal variation of what's inherited by normal offspring. I've seen numbers of about 25 or 50 mutations acquired from father and mother to son or daughter. But that's just the iPSC. But the reprogramming also likely will induce hundreds of mutations. Have you looked at, with your process as it currently is, how many mutations are acquired during that differentiation step?
Yeah, so we've done, so also in the church lab, I developed some whole genome sequencing quality control for stem cell lines. This was a paper called Seq Verify.
Mhm.
And I mean, we've been doing this analysis on our stem cell lines as well. We haven't seen anything that would indicate this is going to be unsafe. So I don't think that will be a major issue here. And I will say that our technology, which uses these like regulatory factors to directly drive um, the developmental transitions that we need, compared with other labs that are trying to do like a more um, a signaling approach, so using the exact same hormones that are found in the natural ovarian development. Our method is much faster, and this has advantages not only in terms of like speed of the experiments, but also it means there's less time for cells to accumulate mutations during that developmental process. So we think that I think we're in a quite good position there with respect to safety, because our method is faster than others that might require more prolonged culture.
Yeah, those two goals go together with the company, both
Yeah.
speed and safety. And so back to that high level overview from the moment the patient walks in to donate a cell to getting an egg back, how long, what are you targeting, and where do you think you are now?
Yeah, so I think end to end. So from a stem cell, right?
Mhm.
Um, Let me think. The thing is, we've recently been developing a lot of different approaches, some of which are faster than others. I think our most likely estimate is on the order of about 6 months, at least with our current technology. And this covers multiple stages, including the like reprogramming, the meiosis, the follicle culture, things like that. Um, I think, yeah, probably around 6 months, although there may be room to get it even faster, um, is what we're looking at in terms of the end to end process. And again, this allows you to produce like as many eggs as you want. Um, and I will say the number that I said, 6 months, could change significantly based on how the technology develops. But I would say like that's around what it's looking like right now to um, my best estimate.
Excellent. And what type of long-term safety studies would you want before moving into clinical trials? You mentioned NHP. Are you looking for multiple generations or just the first generation? What's your safety roadmap look like for that? Or independent of the FDA, what would you want to hold for your product?
Yeah, so I think so. In terms of FDA requirements, I think they are going to require one generation, right? So you make an NHP baby from your stem cell derived gamete, and then that like non-human primate grows up and is able to be fertile on its own, right? And the second, the monkey's child might not need to grow up all the way, just showing that it's fertile is
Yeah.
the important part. Um, that, depending on the species of monkey that you use, will take maybe slightly over a year to up to 3 years. Um, and I think like that's basically around in like what we would want to see as well, in order to know that our method is safe. I think
Mm-hmm.
having the non-human primate at least reach adulthood and see that there's no like developmental abnormalities that might become apparent later is going to be important. But besides that, that plus the human embryo quality control data, I think will be sufficient for us to be comfortable moving this into clinical trials.
Yeah, I want to talk a little bit about the timing. As you know, and I'm sure the world knows, the fertility rates and reproduction rates are going down in the world overall, and in the U.S. particularly. Um, did this inspire you to help create this company? And what do you think might be some of the underlying causes, and how can OVEL help with that in the future?
Yeah, so in terms of like global fertility rate trends, I actually think a lot of this is more due to economic factors like cost of housing, cost of childcare, things like that. I think that many people are not able to have the kids that they want because they wait until later in life
Mm,
in order to start trying. Um, and OVEL could help with that. And I think it will have a quite big impact in sort of extending the reproductive age range. Maybe not like a huge amount, but I mean, I guess like enough to make a. Sorry. What I'm trying to say here is that I think OVEL's technology will make an impact, but the main forces that are driving the fertility drop are sort of more economic than biological at this point.
yeah, and you mentioned it probably could extend the age range a little bit. Very exciting field of epigenetic reprogramming that with those same Yamanaka factors, the tissue is rejuvenated. Are you trialing with iPSCs that are around like peak fertility age, or have you tested a wide range of ages with your technology to see or support that it may support older donors?
Yeah, I would have to actually look up the donor age range of our iPSCs, but I think at least one of our lines is from a woman of like menopausal age. Um, yeah, we get, I think that there's, in terms of the ability of these lines to make eggs, they'll be pretty equivalent. I think in terms of like what society accepts in the age range of women reproducing, like there have already been women up to age 70 who've
Yeah.
had babies with donor eggs, right? And the uterus is still functional um, for quite some time after menopause. So I think, yeah, generally, like I don't have any personal reasons why we wouldn't allow women of any age to use this. I think it will have to be something that society has a discussion about in terms of what's appropriate, right? You don't want to get into a situation where um, a woman is actually too old to care for her children properly, basically. But I think, as you mentioned, like this partial reprogramming stuff, like other anti-aging technologies, maybe like in the future we'll just all be living longer, and then it won't really be an issue. Hopefully, we'll get to that point, but I think society will need to figure out what is going to be the norm here, basically.
And one topic that's been raised by some other competitors, such as conception, is the opportunity for gay men to have offspring. Have you worked with male iPSCs and tried to make an egg with that? Or what are your thoughts or plans for that?
Yeah. So personally, I'm supportive of this as well. I think our first focus needs to be on women just because the regulatory path is much clearer in terms of approval. I think once we get that approval, we can then sort of branch out and potentially work on this in the future. In terms of the biology, we have been using some male cell lines as sort of controls for certain aspects here. There's actually some rather interesting stuff that goes on with X chromosomes in germ cell development and meiosis. So males have obviously XY, females have XX. There was a paper from Katsuhiko Hayashi's group where they were able to take XY cells from a mouse and delete the Y chromosomes, and then some of the cells will actually just spontaneously gain another copy of the X chromosome. It's not very common, but they can select ones that do have this. So in effect, they can convert an XY cell line into an XX cell line, and then they were able to make eggs from that. Um, a similar process could work in humans. There are some differences in how mouse and human X chromosomes function in terms of their status in stem cells. So it's not. It will be interesting to see if the same approach is directly usable. My guess is it might be. It might require some additional optimization. Um, but yeah, I think scientifically this could work. In terms of the regulation, right now anything that introduces a heritable genetic modification, at least in the US, is not allowed. And this would fall under that sort of category. So IVG, which, assuming we use something like mRNA, which doesn't permanently integrate, um, is actually currently allowed, but something like this from an XY starting point would not be. But yes, I think over the long term, I would like to see this happen. It's just right now we need to focus on the sort of core problem, I would say, which is expanding female fertility.
Interesting. And you brought up something of in US current guidelines don't allow for heritable modifications or germline modifications, but we accept some level of random mutations, right? Just from the cell source and the reprogramming. I guess they're not intended as part
Right.
of it, but uh, passengers that go along. Do you think that'll cause a regulatory issue, or
Well,
they'll accept
if
that?
you ban random mutations, you ban natural reproduction as well. So I don't think that's going to be um, put into place.
Yeah, I guess natural isn't the therapy, but current IVF
Yeah.
already accepts those random mutations. Okay, and just to wrap up the ethics section, what are there any other ethical questions that have confronted you while working on OVEL that you'd like to discuss or that are on your mind?
Yeah, I think basically just how are we going to make sure this technology is accessible to everybody? And I think the sort of inequality argument is that, like, okay, if only like very rich people can use this and it allows them to do like better embryo selection and have a genetic advantage for their kids, like what effect is that going to have on society? I think the answer is we need to make our technology like as good and as affordable as possible, so that like it actually comes down below the cost of IVF currently. And I think at some point, like health insurance will cover this, at least in the U.S., and hopefully national health systems of other countries will also be able to cover this, because I think over the long term the benefits will become quite apparent um, of this approach.
Yeah, and what, just to switch a little bit more, what about in vitro spermatogenesis? Do you know of any other groups doing this, or
Yeah,
how did you think of?
there's a company called Paterna, um, who I think I might not be like entirely up to speed on this, but their approach is actually not to grow the sperm in vitro, but to make spermatogonial stem cells and then transplant those into the testis of an infertile man, and the stem cells will then repopulate it and be able to make sperm from there. I think in vitro spermatogenesis has sort of lagged behind a bit. There's a few reasons. One is that sperm are already cheap. So, I mean, it's easy to get 100 million sperm. It's not easy to get 100 million eggs. Um, the other thing is actually the testis, in terms of development, has more like long range organization in terms of like seminiferous tubules than ovarian follicles have, such that if you want to make an artificial ovary, you basically just need to randomly mix together ovarian supporting cells and egg precursor cells, and they'll sort of self-assemble into follicles, right? If you do this with a testicular organoid instead, you'll basically get like some short range organization, but not the long range tubule structures that you actually need in order to make fully mature sperm. So there have been some mouse studies that have made like immature sperm that has actually been able to make live mouse offspring from this. But I think the biology is there are different challenges. I'm not going to say it's easier or harder than making eggs, um, but the technology making eggs is not sort of directly applicable. And then also just there's less there's a less pressing need for this clinically. But I think like our technology in terms of what we're doing at OVEL could easily be applied to the spermatogenesis question as well. So perhaps we would end up licensing it to another company who's working on this.
Okay. And you've brought up embryo selection or germline engineering a couple of times. You've also referenced before there's currently a moratorium for this research in the US, but there's been lately a recent bout of interest. Sam
Mm-hmm.
Altman, OpenAI CEO, has publicly supported nucleus genomics towards embryo selection. Other companies like Manhattan Genomics have come out just to kind of bring into light germline modification. What are your thoughts on how we can ethically go into this into the future, or just tell
Yeah.
us your point of view?
So honestly, I think right now, selection is actually more powerful than editing for the traits that most people care about. Like if you want to make um, certain knockouts like PCSK9 for heart disease, right? You can there are a few genes where knocking them out will have beneficial effects, but there's actually not that many of them. And if you want to think about other things like cancer, Alzheimer's, just other health traits, um, we have actually pretty good polygenic scores for figuring out like okay which genetic variants are correlated with this phenotypic trait, such that we can use these scores to do embryo selection and pick the embryos with the lowest, for example, disease risk. Where that sort of breaks down is if you try to do editing, because what these polygenic scores tell you is which variants are correlated with your trait, and oftentimes there's like several variants that are genetically linked. They're close together on the chromosomes and inherited as a group, right? And one of them might be the one that's actually doing something, but if you do this genome-wide association study, you'll find like all of them in the group sort of indistinguishably have the same effect, which means that if you try to do editing, you don't know which one to edit. Um, so basically, that's why I think selection right now is more stronger than editing. As editing improves and as our understanding of the biology improves, this could change. But also, I think just in terms of the regulatory environment, the selection is better. The other thing is, if you do embryo editing, you get basically one chance to get the edits right. And as we saw with the CRISPR babies in China, um, it doesn't always work out how the scientist intends. And yes, you can do some amount of quality control after the editing, but you might miss things, especially if the editing was like partial, only present in some of the cells. With like if you're thinking about editing, and let's say for the sake of argument that this does become legal in the U.S. in the long term, right? I think you're actually better off doing your edits in a stem cell line and then doing IVG on the stem cells to make the eggs, because at that point you can verify that all your edits worked. There's no unwanted or harmful off targets, things like that, before you go and actually make an embryo from it. Again, I think this will need to be regulated in order to make it safe, in order to make sure it's not like abused or anything like that. But I think that IVG has a place even in things where you're considering embryo editing. Regarding like the recent burst of interest, I'm still sort of, well, I'm not directly involved in any of this. It's going to be interesting to see how it plays out, because right now, as I mentioned, this is not currently allowed in the U.S. So I'm sort of curious to see what their strategy will be. But maybe when you're Sam Altman or Brian Armstrong, you have some leverage to potentially get the regulations changed. We'll see.
And they've discussed about just going to other countries. I think that's the short-term solution. The
Maybe.
research may happen here, but early trials, if they can't change the policies, would be to go elsewhere that would support
Yeah,
it.
yeah.
And wanted to switch ways to the trend of AI in discovery and in drug development. Is OVEL using AI, and if so, in which way?
Well, we've been certainly using a lot of like more like machine learning approaches that don't like that are not what people think of when they hear AI these days. For example, like automated image processing, segmentation, things like that. Like neural nets, but not like transformers. We have also been using Gemini quite extensively in terms of things like literature search. Like it's pretty good for literature search. It's definitely saved me quite a lot of time. Also for coding, our analysis pipelines, things like that, um, and some data analysis as well. But I have not found it to the point where it's been able to do a better job analyzing and interpreting data than one of our scientists would. But it's definitely saved us quite a lot of time on literature search and coding. Going forward, I think like the main thing that AI needs to succeed is training data. And there's actually like not a ton of high quality stuff. Like in terms of single cell sequencing, there is good public data. In terms of other stuff like chromosome morphology or like certain epigenetics data sets, like I think AI might be more limited if we don't have the data accessible, and we're in the process of generating that data, um, but there have been times where like I've asked, like we have a Gemini Pro subscription, right? I've asked Gemini Pro to figure something out, and it comes up with the same idea that I did, that I'm asking for its help with because that idea did not work. And actually, occasionally it's said like, "Hey, have you checked out this paper published by Dr. Smela on this subject?" It's like, yes, this might be a relevant paper. Thank you.
It's pretty funny because usually it knows who you are, at least when you're interacting with these. I don't know if you've shared that, but
Maybe.
funny that it gave your own paper back to you. So we're getting up on time, but wanted to close with some science fiction. You mentioned that it's influenced you in your early story. Could you just go into a little bit about how science fiction has influenced you and how you think it has influenced science broadly?
Yeah, well, I think like it's influenced me in several ways. One is like just being able to imagine what sort of worlds might be possible as technology develops and which worlds we want to aim for. I think, I mean, one thing that gets mentioned a lot is something like Brave New World, right? Where humans are grown in vats, society is like highly regimented based on like whether your vat was given alcohol to like stunt your brain development. They've like messed up a lot, I would say. The question is like, how do we use the technology in a beneficial way instead of a dystopian way, right? And I think that is a question that this sort of science fiction forces us to think about. The recommendation that I gave, or you asked for a science fiction recommendation, there's a Ted Chiang story called The 72 Letters, which has, as part of its theme, I'm not going to spoil it, but basically engineering to like improve reproduction in a way that benefits society. So I think stuff like that is what we should be aiming for. And also, like another thing I've been thinking of, like, is how are humans going to adapt to AI? Right? I think that over the long term, this will benefit from things like there's just a lot of issues in the world right now that ultimately come down to the fact that we have all these heritable diseases, right? Like something like Alzheimer's, right, is in large part genetic. And if over the long term we can have technology that means people aren't at a high risk of this, I think that has a really big benefit to society, um, as as it gets more adopted. We just need to make sure it's in a way that's responsible, in a way that benefits everybody, instead of ending up in a situation where like only certain people can use it.
I love your interpretation that science fiction helps us pick between the dystopian and the proper uses, because it seems modern science fiction shifted more towards the dystopian, but that equally trains your mind of
Yeah,
what not to do. Everyone thinks of Jurassic Park and
or
the
or
woods of
the
what.
torment nexus, as people like to say these days.
Yeah, very interesting. I love Ted Chiang. I've read several of his, and
Yeah.
I'll have to go and check out that particular short story. Have you read the one of Ted Chiang where a treatment makes people super intelligent? And it's the
Yes,
story of
understand.
understand. Yeah, I
Yeah,
love that story.
that's
That's my favorite short story of his. Well, excellent. Let's close out with one of just one part of the show is how we balance these cutting edge new technologies and high passionate work with balance and life. And some people meditate, some people go out and work out, some people are just solely motivated by their work. How do you maintain balance while achieving these great things?
Yeah, well, I will say I'm very grateful for my wife, Ursula, for helping support me in all of this. Um, I think that honestly, like, this is what I love to do. And, like, even in high school, I did science as a hobby. So, I mean, I don't really have a lot outside of this. But, I mean, that's what I'm passionate about, right? Um, so hoping to be able to help all the people who need to have kids, achieve their dreams.
Excellent, well said. And you're very focused on that goal with OVEL, but I'm sure you're well read outside. Are there any topics or new areas in science that have piqued your interest, but you're very committed to the current path of in vitro gametogenesis?
Yeah, in terms of other like biotechnology stuff, um, there's a few things that I've sort of been thinking about off and on. One is like the fact that there's all these like viruses, in particular like Epstein-Barr virus, CMV, things like that, that infect a lot of people and may cause like long-term negative effects, but we don't have any sort of vaccines or treatments for. So being able to fight those would potentially help with like a lot of different things. Like for example, multiple sclerosis, right? It actually turns out it's this virus that causes it. Um, yeah, so I would say like underappreciated common pathogens that like might not make you very sick, but because so many people have them on average, like they have actually quite bad effects. Another one is like just um, I've been following gene drives for a while and the ability to prevent insect-borne diseases, things like that. So how can biotechnology help people like in developing countries who are struggling with these issues? How can we make sure that this actually works and goes well? I would say like those two things. I guess also like um, just been interested in the history of like how we came to common laboratory practices. Like I wrote, for example, an article a few years back on the history of micropipettes or parafilm or things like that. It sort of shows interesting historical choices that shape the tools that we use and how those tools are actually like very underappreciated, I would say, in terms of their scientific impact. Like nobody got a Nobel Prize for a micropipette, but I'd say it had more impact than many Nobel Prizes out there.
Mm. Yeah, well said. Interesting. And to close out, what advice would you give any young scientist who's getting into the field, they're starting their career early on, how to be successful or how to pursue their passion?
Yeah, I think being able to pick a problem, like pick a good problem and then stick to it, right? So I read a biography of Yamanaka, um, and one of his slogans was "You need vision and also work," right? So
Yeah.
VW. Um, so in terms of vision, like picking a problem that you have at least some hope of solving, and that also has a high impact, right? And that's actually pretty hard because often like there's a lot of other people working on it already, so your personal impact might be low. Other problems are like a lot more difficult. For example, this is why I'm not working on artificial wombs. So a uterus is actually much harder to engineer than ovaries. I think artificial wombs would have an even bigger impact, but I don't think it's as tractable. So finding the right problem and then just focusing on it and putting in the work, getting good. Um, I think being able to adapt when things, when like to recognize when a particular approach is stuck and be able to choose a different approach to the same problem is also quite helpful. And I think honestly, like it's hard to learn this outside of just doing it and getting experience. So I would say just think about what problem is interesting and impactful and something you can actually do, and then just get started on it. And yes, you'll run into issues, but eventually you'll get enough experience to identify good approaches.
Excellent. I love VW, vision plus work.
Yeah.
Definitely had some students that have great vision but don't have the work aspect,
Yeah.
and vice versa. You need both. But it's been a pleasure talking with you today, Merrick. I really appreciate it. Thanks for joining the show, and have a great day.
Yeah, thanks for having me. Bye.